So today I'm going to discuss about pharmacokinetics. The definition of it is what the body does to the drug, okay? It's the study of the disposition of a drug. It includes absorption, distribution, metabolism, excretion. And in our course, pharmacy, we can also include in the beginning, like what I've mentioned before, we can also include your liberation and then also your toxicity. Okay? So, pwedeng ADME, and then may toxicity pa sa last. So, this is a simple diagram, a flow chart of what happens in your pharmacokinetics and pharmacodynamics. You already know this one. Focus tayo ngayon sa pharmacokinetics to your ADME. From your drug administration or liberation, and then going to the blood and then to the target site, or it can be metabolized and then excreted na. Okay? For your pharmacodynamics, that includes your pharmacologic effects, clinical response, it also discusses the adverse response and the therapeutic effect. Now, you have here, paulit-ulit lang class, absorption, distribution, metabolism, and excretion. And let's start each one of them. First, we go to absorption. Okay? So, layman's term, yung mabilis maintindihan, absorption, the drug is absorbed inside the body. Okay? So, we have here some factors to take note or some considerations. Pag iniisip natin ng absorption, we think of yung drug ba na transport sa GIT ng maayos? And then also, we have the effects of pH on drug absorption. What are the effects? Physical factors also. Okay, for transport of drugs in the GI tract, we have general principles. First, you must always remember, a cell membrane, it's a semi-permeable structure composed primarily of lipids and phosphate. So remember class, yung cell membrane, it's a phospholipid bilayer. Okay? So magdedepende yung absorption nyo doon sa structure ng inyong drug, kung pasok ba siya doon sa phospholipid bilayer. And then second, wala siya dito. Your transport of GI tract involves transport mechanisms such as pwedeng passive transport, pwedeng active transport, facilitated diffusion, etc. I will discuss later. And then next, usually your proteins, drugs bound to proteins and macromolecules do not easily cross cell membranes. Okay? Since malalaki yung molecules ng proteins, hindi sila madaling makapasok. That's why you have some drugs kasi na bound to proteins. And these drugs bound to proteins, hindi sila yan mabilis na makakapasok sa cell membrane. So always remember that. That's a general principle. Your proteins do not easily cross cell membranes. That's why drugs prone to protein binding, hindi sila mabilis ma-absorb. Okay? You have a lot of drugs that are protein-bound, highly protein-bound. Next, your non-polar lipid-soluble drugs, they traverse the cell membranes more easily than do ionic or polar water-soluble drugs. Okay, class? Since phospholipid bilayer siya, it's a lipid layer. May lipid layer component siya. So, syempre, yung lipid-soluble drugs, sila ang mas nagtra-traverse na mas mabilis. Okay, and then yung last kanina na principle, your low molecular weight drugs, they are more easily absorbed. Of course, class, low molecular weight, mas mababa yung particle size nila. So, syempre, mas mabilis silang makakapasok. They are easily absorbed. I have presented 5 principles, okay? So, yun ang kailangan yung mga tandaan when you think of transport of drugs in the GI tract. Okay, so it shows again the slide we missed before. Remember class, non-polar lipid-soluble drugs, mas mabilis ma-absorb. Okay, so we have here your transport. It depends on transport mechanisms. You have your passive diffusion and partitioning, and also the other one is facilitated diffusion and active transport. Okay, I mentioned to you passive diffusion, facilitated diffusion, and active transport. So when you say passive diffusion, you move your concentration. The drug moves from a region of high concentration to one of low concentration. So remember that class ha? High to low. Wala siyang carrier. Hindi niya kailangan ng carrier kaya hindi siya saturable kasi wala naman siyang kailangang isaturate na transporter or carrier. And then, only the non-ionized form of a drug diffuses across the lipid membrane. The more lipophilic the drug is, the faster is the diffusion. Remember class, pag non-ionized and lipid-soluble, mas mabilis siyang ma-absorb. At equilibrium, the concentration of the non-ionized form is the same on both sides of the membrane. For your active transport, it involves specific carrier proteins that span the membrane. And kailangan niya ng energy to transport the drug. So remember, active transport has a specific carrier para matransport yung drug. It is driven by hydrolysis of ATP. The drug is capable of moving from low to high concentration. So tingnan niyo lang dito sa picture, you have passive transport and active transport. Your active transport, it involves a carrier. While your passive transport, okay, ano namang effect ng pH mo sa inyong drug absorption? So, if you have a drug that is uncharged, it is readily soluble. Yan. So, a non-ionized or uncharged drug is more lipid-soluble and it diffuses across your cell membrane more easily. Kasi your cell membranes are meron silang electrical property. So, by nature, they are ionized yung membranes ninyo. And ang mas mabilis na makakapasok na drug ay yung non-ionized because they will attract each other. Non-ionized to ionized. Pag ionized na yung drug mo, tapos ionized pa yung membrane mo, walang masyadong attraction. Okay? Kasi ionized na siya bakit pa siya lalapit doon? So, your non-ionized drugs, they are more easily absorbed than your ionized drugs. Okay? Okay. So, permeability of the membranes. Your lipid-soluble and ionized dissolve in the lipid membrane during transfer passive diffusion. May ion pair kasi na nangyari. Now, your water-soluble or your lipid-insoluble substances of small molecular weight transfer through water-filled pores in the membrane. It's called your convective transport. Okay? Lipid-insoluble sila, water-soluble sila, pero small naman ang kanilang molecular weight, so they can still enter. Now, the transport that you need for that is your convective transport using your water-filled pores. Substances may combine with the carrier also. That's for your transport by special carriers. Now, going back muna dun sa passive diffusion, it's a process governed by Fick's law. So, under passive diffusion class, you have your simple diffusion and your convective absorption. Yun sa mga drugs na para sa water-soluble drugs. Now, you have your special carriers, yung active transport nyo, and your facilitated diffusion. Your transport by special carriers, kailangan nila ng magbubuhat dun sa drug or kailangan ma-attach ng drug dito sa carriers na ito. And then others, you have your ion pair transport, and then also you have your endocytosis, exocytosis, and phagocytosis na not so used transport mechanisms. So, ang main transport mechanisms are your passive diffusion, your facilitated diffusion, your convective absorption, and your active transport. So, those four. Remember class ha, you have passive diffusion does not need a carrier. Okay, it is not saturable. Wala siyang kailangang isaturate na carrier, na transporter, na channel. While your special carriers, your active transport needs ATP, your facilitated diffusion needs a channel. Okay, so kailangan nila ng mga carrier ito.
Activity is the ability to move materials into and out of cells. This is important in cell communication and normal cell function. For example, in order for the cells of your nervous system to function properly, ions, water, proteins, and other molecules need to be able to pass into and out of cells. The cells of the nervous system form networks. They, like all cells, are able to function because they can control what substances are inside the cell and what substances stay outside the cell. These materials move in and out of cells by passing through the plasma membrane of the cell. The plasma membrane surrounds the cell and separates the interior of the cell from its environment. This semi-permeable membrane is composed of a lipid bilayer. Phospholipids make up a large part of the membrane and form a bilayer. The structure of the bilayer is due to the tail-to-tail packing of the nonpolar hydrophobic tails, composed of two fatty acid chains, and the polar hydrophilic heads, composed of glycerol and phosphorylated alcohol. The lipid bilayer is 5 to 10 nanometers thick and is embedded with proteins. Some cell membranes also contain cholesterol. A plasma membrane contains different types of proteins which are specific to the particular function of the cell. These proteins also enable the cell to interact with its environment. The entire structure of the plasma membrane can be described as a fluid mosaic model. The phospholipid bilayer has properties resembling fluids, and the differing proteins and their attachments on either side of the membrane resemble a mosaic. The plasma membrane is the gatekeeper of the cell, allowing certain substances in and out of the cell at certain times in certain amounts. Diffusion is a process in which substances move across a membrane from an area of high concentration to an area of lower concentration, or between areas of opposite electrical charges. This is called the electrochemical gradient. Small, non-charged particles, primarily gases such as oxygen and carbon dioxide, can diffuse through the plasma membrane by moving in between the phospholipids in the bilayer. However, the cell needs to control what enters and leaves, and so transport proteins aid in the selective movement of other molecules across the membrane without the input of energy. Through facilitated diffusion, larger molecules, polar molecules, and charged ions use channel proteins embedded in the bilayer. The transport of other substances requires a special carrier that will bind the substance on one side of the membrane, which triggers a conformation change in the protein carrier, causing it to release the substance on the other side. When the substance being moved across the membrane is water, the process is called osmosis. The cytoplasm of the cell, as well as the interstitial fluid, is composed of solutions. The solvent, usually water, moves across a semi-permeable membrane toward a higher solute concentration, consisting of various molecules or ions, until equilibrium of the solutions is reached. The plasma membrane contains proteins called aquaporins, which are specialized channels for the movement of water during osmosis. A cell in a hypertonic environment will have water move from the inside of the cell toward the higher concentration of solutes in the solution outside the cell. In a hypotonic solution, the concentration of solutes is higher inside the cell than the outside environment, so water will diffuse into the cell. When the solutions on either side of the membrane reach equilibrium, they are referred to as isotonic. When substances need to be driven against their concentration gradient from low to high concentrations, active transport is required. This process uses energy to move a substance across the membrane using selective protein carriers. The sodium-potassium pump is a specialized membrane protein carrier, common in most animal cells, that moves sodium and potassium ions across the plasma membrane. ATP fuels the pump in the movement of these ions from low to high concentrations, moving sodium ions outside of the cell where they become concentrated and bringing potassium ions into the cell where their concentration is higher relative to the outside. In other instances of active transport, the concentration gradient created from the action of one pump can be used as the input of energy to drive a different pump. a process known as coupled transport. The large concentration gradient of sodium ions is used to bring glucose into the cell against its concentration gradient. The plasma membrane, with its lipid bilayer and various embedded proteins, provides a barrier between the internal cellular environment and the environment surrounding the cell. The structure and function of the plasma membrane, coupled with the processes of moving materials into and out of cells, allow for the metabolic action cells need to function, as well as for the continuation of life.
Okay, blood flow to the absorption site. So your blood flow also influences your absorption. Now, blood flow is greater in the intestines than in the stomach. That's why in the intestine, your drugs are greatly absorbed. The blood flow to the GIT is important in carrying the absorbed drug to the systemic circulation. Now, your splanchnic or mesenteric circulation receives about 28% of the cardiac output. So, ang laki, out of 100%, 28% ang napupuntang blood flow sa inyong intestines. Kaya maganda ang absorption doon. Now, your lymphatic circulation, pwede rin magkaroon ng absorption ng inyong lipophilic drugs and dietary lipids in the lymphatic circulation. Maganda siya kasi it bypasses the liver. Now, for the total surface area available for absorption, magandang absorption sa intestine because mataas ang surface area niya because of the presence of microvilli. In your skin, less ang absorption because 1.73 meters squared lang ang kanyang surface area compared to your GIT na umaabot ng 200 meters squared. You have a part in your GIT, a segment, where the drug is well absorbed and you call it the absorption window. Well, beyond that absorption window, the drug is either poorly absorbed or not absorbed at all. And that absorption window is at the proximal area. It has a large surface area. It is well supplied with blood and it maintains a large drug concentration gradient favorable for drug absorption. So, sa duodenal yon, proximal area. Contact time is also important. When the drug moves through GI tract very quickly, hindi siya na-absorb ng mabuti. Okay, another factor that will affect your absorption, the bioavailability of the drug. It is the fraction of administered drug in a chemically unchanged form that reaches the systemic circulation. Remember, your bioavailability is the rate and extent of drug absorption. How do you assess it? You compare the plasma levels of your drug after a particular route of administration with the plasma drug levels achieved by IV injection. In bioavailability class, you have two types. You have the relative bioavailability and the absolute bioavailability. So how will you differentiate the two? When you say apparent or relative bioavailability, it means the availability of the drug is measured comparing it to the recognized standard. So, the availability of the drug from a drug product as compared to a recognized standard. Iko-compare mo yung AUC ng drug na ito dun sa AUC ng standard na drug. Okay? So, you call it relative or apparent availability. That has a formula class. In the future, I will be showing you how to compute pag napupunta na tayo sa mathematical calculations. The relative bioavailability of two drug products given at the same dosage level and by the same route of administration can be obtained using the following equation. So, hindi niya pinapakita dyan, pero it's very simple. You just divide your AUCA over your AUCB. Your AUCA, that is your area under curve. Okay, ayan na. That's the area under curve for your drug that you're going to test, and then your AUCB, that's the AUC of the standard drug. Next, absolute bioavailability. It's the systemic availability of a drug after extravascular administration and then compared to your IV dosing. So, kung ang relative kino-compare niya ang isang drug and then i-compare niya sa standard drug, ang absolute bioavailability naman, it's the term given when you compare your oral route of administration or other route, any route, and then you compare it to the IV dosing. Ko-compare mo yung AUC ng dalawa. Okay? It's just the same drug. It's isa lang ang drug dito na pinagko-compare. And then you compare their bioavailabilities by their route of administration. You compare its bioavailability gamit ang iba't ibang route of administration. So that's your absolute bioavailability. Okay, the absolute bioavailability or the F value is equal to AUC oral over AUC IV. Okay, so what if you are given a question? For example, what is the relative bioavailability for an experimental drug tablet versus solution or control based on the following data? So your drug tablet, that's the one, your experimental drug, that's the one that you're going to test, and then you will compare it with your standard drug or your control. So dyan, makikita nyo lang tablet and solution, yun yung control. So you divide your AUC 20 over AUC 30. Okay? And then you multiply it by 100% to get the relative bioavailability. So remember class, relative bioavailability, you compare the experimental drug versus the control drug. Okay? So next. Okay, so this is your assignment class. You solve it on your own. You solve for the bioavailability and then you interpret it. Is it less bioavailable? Is the oral administered drug less bioavailable than your IV? Okay, a drug given by the IV route will have an absolute bioavailability of 1. F equals 1 or 100% bioavailable. While drugs given by other routes usually have an absolute bioavailability of less than 1, like your oral drugs. Yung kinompute natin kanina. Now, this is a graph comparing your AUC oral and your AUC IV. So, pansin nyo, mas malaki, mas mataas yung sa AUC IV ninyo. Okay?